In 1910, Thomas and Morgan discovers an unique genetic inheritance pattern in Drosophila, which is completely different from Mendel's observation. And this unique genetic inheritance pattern is now known as linkage. Hello friends, welcome to Biology Concern. I am Animesh, and in this video I am going to discuss about linkage, that is the genetic linkage, coupling, repulsion, and all possible factors of linkage. So let's jump into the topic. Firstly, the discovery of linkage was made in 1910 by Thomas Hunt Morgan. So Thomas Hunt Morgan discovers this unique pattern of inheritance in 1910 in Drosophila melanogaster. And now we know that this linkage is an exception of Mendelism or Mendelian genetics. So basically, these inheritance patterns are completely different from Mendelian inheritance pattern, that is monohybrid cross or dihybrid cross. So what is the linkage? By definition, linkage is referring to the close association of genes on a chromosome, and that close association of genes are inherited together in offsprings when a gamete is formed. So basically referring to the close location or co-location or association of genes or DNA variations to each other on the same chromosomes, and the closer the genes are to each other on a chromosome, the better chance to detect the linkage, because they are then more likely to be transmitted from parents to offsprings. So if we consider this is a chromosome, okay, so this is a chromosome and this is the centromere. So if we consider this is a gene A and a gene B, and in its opposite or homologous chromosome, this is a gene small a and this is a gene small b. So whenever there is a gap present in between the genes, so whenever there is a gap present in between the genes, then the chances of crossing over are greater. So in this case, the gap present, okay, between the genes, okay, so in this case crossing over, the possibility of crossing over is higher. So crossing over possibility is higher, okay. But let's assume that this is a chromosome and this is its homologous chromosome, okay. So let's assume that capital A, capital B and capital C, those three genes are present in close association with each other in the chromosome. Say for example, this is chromosome 1 from father and this is chromosome 1 from mother, okay. And in the case of mother, here small a is present here, small b is present here and small c is present here. So the genes are present here in the mother chromosome or the chromosome of mother. There is a distinct gap is present. So a distinct gap is present between the genes, but in case of father there is no gap, there is an absence of gap. So in this case, absence of gap. So in here genes are closely associated. So genes are closely associated with each other. So in this case, so there is the least possibility of crossing over. So crossing over does not occur here. So in case of here, crossing over is not possible if there is a less frequency of crossing over, then these genes should inherit together in the next generation. So whenever from P1 to F1, so from P1 to F1, these genes are likely to be transmit together in the next offspring. Okay, so these genes are likely to be transmit, so these genes that is closely associated with each other are likely to be transmit with each other in the next generation. So whenever there is a close association present between the genes and there is a less frequency of crossing over, then the genes inherit together from parent to offspring, and this process or this phenomenon is now known as linkage. So the linkage, the probability of linkage, so the probability of linkage is 41.5%, 8.5%, 8.5%, and 41.5%. So these 40 Morgan observed that there is a probability of 41.5%, 8.5%, 8.5%, and 41.5%, and this is totally different from Mendelian inheritance. And so this is totally different from Mendelian inheritance. Okay, so in Mendelian inheritance we observe 9:3:3:1, but in case here 45:8.5:8.5 into is to 45.5. okay, so there is to, so this probability of linkage are definitely different from the probability of Mendelian inheritance. Okay, so these are the linkage. Now two more factors are concluded in here, that is the coupling and repulsion. So what is the coupling and what is repulsion? So when we consider two of the genes, one is dominant gene and another one is recessive gene. So if dominant gene is A and B and recessive gene is small a and small b, then in homologous pair of chromosome, if A and B are present in a same chromosome and in other homologous chromosome small a and small b is present, then this position or this organization is known as coupling. So this kind of organization is known as coupling. And if capital A along with small b and small a along with capital B, that means one dominant gene and one recessive gene present together in a same chromosome and the dominant gene are present opposite in the homologous chromosomes, then this kind of organization is known as repulsion. Okay, so this kind of organization is known as repulsion or trans organization of chromosomes or genes, trans organization of genes. So basically in linkage, close associated genes are transmitted together in parents to offspring, and whenever those dominant genes are present in same chromosome, then this kind of organization known as coupling, and whenever the dominant genes along with recessive gene present in the same chromosome, then this kind of organization is known as repulsion. So I think this video is helpful for you to understand the genetic linkage. If you like that video, please hit the like button, don't forget to subscribe, and thanks for watching.